Dr. Annelise Bocquet, welcome to Weird Sauce, and thank you for making time for us today in your very busy schedule in France. Could you kindly introduce yourself for our audience, please?
Yes. Well, my name is Annelise Bocquet. I'm a PhD in biology. I obtained my PhD in the end of 2012, and I work on microglia cells activation in an animal model, the leech Iridomedicinalis. This invertebrate can regenerate its nervous central system after lesion, and microglia cells are involved in this phenomenon. I'm specialized in immunology much more than in neurosciences. Well, I really apologize for my awful English accent, and I will do my best to be understandable. And thank you for inviting me to this podcast.
You're most welcome. It's been a very long road for people who started the story of COVID and since the 5 years that we've been in this. And I think you've been one of the earliest voices that I could find online talking about very complex things and trying to unravel the subject. And we know the subject was actually much more complicated than we first thought.
Right.
And therefore, knowing that we're now in the latter part of 2025, from your experience in terms of both having started to look at it from the onset, so looking at the virus and looking at the vaccines and the various evolution of people who got vaccinated, who got the virus, and cross infections and so on. I would love for you to be able to give the audience a sort of panel of where we at this junction in terms of this enormous event that's happened to humanity.
Okay. Um, well, I've been working on the immune response induced by the SARS-CoV-2 spike protein for 4 years now. And why? Because this protein is present on the virus and is produced by our body cells after injections of anti-COVID-19 vaccine, the so-called vaccines, products such as those from Pfizer or Moderna. And I published a first peer-reviewed paper on queries entitled Impact of the SARS-CoV-2 Spike Protein on the Innate Immune System, a review. And now I'm more interested in the immune tolerance mechanism in which the SARS-CoV-2 spike protein is involved. Why? Because we have to understand that this immune tolerance mechanism can explain the asymptomatic COVID-19 cases. And it can also explain its persistence in our body. The fact is that the immune tolerance mechanism render our immune system ineffective at eliminating the virus and its spike protein, or the spike made after the administration of anti-COVID-19 products from our body. And today, we know that spike is detected during many months after an infection by the virus, but also after anti-COVID injections in the blood. And it probably explains why we have long COVID cases and we have some post-vaccination syndromes, also called PVS. Okay? I can quote some papers like Schwank et al., Yonker et al., Zui Wang et al., or Nakao Ota et al., or Bhattacharjee et al. To support my words. Okay. So the immune tolerance mechanism can explain the persistence of the SARS-CoV-2 spike protein over time. Okay, but it is insufficient. It's not enough to explain all the phenomena, right? In fact, spike can be continuously produced by our cells after an infection by SARS-CoV-2 or after administration of modified mRNA formulated, that products are formulated to persist in our cells. Okay. One of our biggest challenge today is understanding how the virus SARS-CoV-2 can persist in our body on the first part. Okay. Or how the modified mRNA can persist also in our body. That these two parts are linked. And I think that the spike protein produced after administration of anti-COVID-19 product can allow the viral persistence too. In short, I think that the mechanism induced by SARS-CoV-2 spike protein can be one of the explanations, in fact. In particular, by triggering cell survival mechanism in infected or in transfected cells. I can cite the mTOR activation described in Linley Xu et al. Preprint, or p53 inhibition described in Milani Daniela et al. Paper.
You may want to explain what P53 is.
P53 is the gradient of our genome. It's a protein our cells produce, and it can be activated in case of cell damage. All right? Is that enough?
Yep.
Okay. So we had to apprehend the survival processes of these cells infected by the virus, SARS-CoV-2, or having received modified mRNA. And I think spike is involved in these processes. Spike, in fact, spike can activate mTOR. Okay, it's an activation that induces cell survival processes, or can interact directly with p53, our guardian of our genome. Thus, spike can maintain its production by keeping infected cells or transfected cells, that those cells that have received the modified mRNA, alive. Okay? Is it? Do
Yes.
you understand?
Yes. So I think for the audience, because if you're listening to this rather than watching Annelise's expression, if you're just listening to this, she means to reinforce how meaningful and how incredibly important this particular aspect is. And I think if you understand the situation in public health in various countries right now, and you look around in the general health of your families, your friends, etc., you may be able to understand and translate what she's saying in terms of scientific terms into what you're observing around yourselves.
Okay. So, in summary, we have two different ways to explain the SARS-CoV-2 spike protein persistence. First one, the immune tolerance mechanism. Second point, its continued production via virus reservoirs or transfected cells kept alive by spike. Okay? The major problem today is that the SARS-CoV-2 spike protein is amyloid, regardless of its origin, right? Since the amyloid sequences are conserved between the spike protein of the virus and the spike protein, the so-called true proline spike protein, uh, that one produced after the administration of anti-COVID-19 products. It's also called spike 2P. So these two spike protein are amyloid.
Can you define what that is, amyloid?
Amyloid, it's just a small sequences in the protein that after the release can clumps itself and form a network, and that network is degenerative. We find this in Alzheimer's disease or Parkinson's disease or even Creutzfeldt-Jakob disease.
It's a brain pathology.
Yeah.
Yes.
Neurodegenerative pathology disorders. Yeah. So, if the SARS-CoV-2 can persist in reservoirs, the spike protein of the virus is continuously produced. And we detected it in the blood. Okay? But it possesses amyloid sequence. Okay? And it can interact with other amyloid fibers and induce their production by our cells. And via these processes, it can amplify its spread in our body. I don't know if you understand really well what I am saying. It's a double amyloid effect. We can see. Well, how can I explain that in a simpler way?
Well, it's a cumulative effect and reinforcing at the same time.
It's much more than accumulating effect. Um, um, we know that spike can activate one receptor. This receptor is named RAGE, a receptor for advanced medication and products. It's a receptor. Okay. This receptor, when it is activated, can release some alarmins like HMGB1. Okay. Interestingly, the RAGE HMGB1 system can recruit anti-inflammatory cells called MDSC that produce interleukin 10. Okay. And this anti-inflammatory cytokine is really of interest because it induces ACE2 production, ACE2 expression. And ACE2, we know that it's the receptor of the virus. Okay? So this phenomenon, ACE2 overexpression, can explain why the virus invades our organism during an infection. Okay? But even more interestingly, amyloid beta 42 fibers, that are fibers, degenerative fibers we can find in Alzheimer's disease, for example, can also activate RAGE and induce the release of HMGB1. So it can activate also the RAGE HMGB1 system with the ACE2 overexpression. Okay? So both spike or A beta, amyloid beta 42 fibers we found in Alzheimer's disease can induce the expression of ACE2, the receptor of the spike protein on the virus. Well, okay. And even when linked to amyloid beta 42 fibers, because spike can interact with amyloid beta 42 fibers, even when linked to these fibers, spike can still interact with its receptor, ACE2. We are therefore faced with a double amyloid effect. Not only is spike amyloid degenerated in itself, okay, but it induces the production of amyloid fibers, including amyloid beta 42, and it enables the induction of immune tolerance mechanisms via RAGE HMGB1 interleukin 10 system that increase ACE2 expression, and it can use amyloid fibers. Therefore, it can use amyloid fibers to spread throughout the body. That's the double amyloid effect of the spike
And
protein.
in your mind, understanding those biological mechanism, what are the likely pathology that we are able to connect with those things right now that we are seeing clinically?
Dementia, we can connect it to dementia. Also, all the impaired all the neurological disorders like Alzheimer. Recently, we have a paper explaining that the spike of the SARS-CoV-2 can induce Parkinson, so Parkinson disease, and also Creutzfeldt-Jakob. That is a really terrifying disease. We know that spike can interact with the prion protein, the human prion protein, and can induce its fibrillation. Thus, it can made the prion protein, the human prion protein, becoming deleterious. Okay, it's quite complicated to explain that in simple words.
No, but it's the important thing is, I think the important thing is for people to understand that there are multiple pathways. They are additive to some extent.
Yes.
They are somehow individual as well.
Today, there are few case reports in the scientific literature describing the development of this kind of pathologies following an infection or after anti-COVID injection. Yes, both.
Yeah, but both aspects. So then we also have the issue that I don't know whether that's actually studied at all. The idea of both having had people were exposed to the virus, then got injections,
Yeah.
and then there's obviously a question of how many injections did they
Yeah.
get. and then may or may not have got reinfected with the virus. So those particular folks, are they presenting with evidence of accelerated impact on the brain versus, but are we even doing this research to your
Yes.
knowledge?
And this cumulative effect, because what you are describing is in reality a cumulative effect by repeated exposure. Um, so um, it's not only induce spike, doesn't only induce degenerative disease. It's also impair our immune system.
Yeah, immune
Yeah.
system. Yeah.
Today, many articles draw parallels between HIV and SARS-CoV-2. What do we know about the implication of PRP, the prion protein, in HIV infection? PRP is a normal protein found in the brain and other tissues, um, particularly on immune cells. Well, I will develop this point a bit later. You have to know that its cellular form, called PRPD, is harmless, but it can misfold into a rogue version named PRPSD, okay, that clumps together, causing disease like Creutzfeldt-Jakob. These are called prion disease, and that damage the brain. How HIV and PRP might be linked? Well, I recently read an article entitled The Role of Shed Prion Protein in the Neuropathogenesis of HIV Infection. This article shows that in HIV positive people, levels of PRP, especially the shed form of PRP, is detected in the cerebrospinal fluid, the fluid around the brain and the spinal cord. And these levels of PRP in the cerebrospinal fluid are altered. Normally, PRPC is a helpful protein, but in HIV, its shading increases due to inflammation caused by cytokine like TNF alpha or CCL2. And this shading might be the body's attempt to protect against brain damage. But it can backfire by attracting immune cells that spread the virus or cause more harm. Both HIV and prion disease cause chronic inflammation in the brain. And the article that I mentioned above earlier explains that shed PRPC in HIV patients contributes to this by triggering cytokines which recruit immune cells and worsen brain injury. I recently suggested that SARS-CoV-2 and its spike protein might do something similar. As researchers found the virus responsible of the COVID-19 disease and its spike protein in immune cells and in the central nervous system. The fact is that HIV also leads to degenerative disease due to misfolded protein. And we know that the SARS-CoV-2 spike protein can mimic or induce directly this misfolding, linking SARS-CoV-2 and spike to both HIV and prion-like effects. I don't know if it is clear, but what I want to say today is that SARS-CoV-2 and spike are linked to both HIV and prion-like pathologies. The altered PRP levels in HIV positive cerebrospinal fluid supports my hypothesis that similar immune and neurological disruptions can occur with SARS-CoV-2 and spike protein. The spike protein from the virus, but also from anti-COVID-19 injections. Okay? And the fact that, and the fact is, if spike can contribute to cell survival processes, keeping infected cells, but also transfected cells alive with a continuous production of the spike protein, we will have to face a huge disaster in for the neurodegenerative disease with an HIV-like infection.
Have you read in the papers that you've read? Have you come across a factor for age, or is that not even a factor? So this would be affecting people regardless in its strength, regardless of the age, or do we have some effect in the older the person, the worse the effect? I'm wondering whether there is an age thing because um, young people with certain type of presenting issues right now
Yeah.
clinically, we can't really make sense of what's happening there, and they're very very young, so they would be very unusual for them to be considered patient with brain aging issue,
Mm.
um, and they're already presenting with some of the substrate of brain aging. So I'm wondering whether you've seen that in the literature as well, that there is a an effect of age or not.
Um, we have some uh, case. We have some case reports of young people suffering after SARS-CoV-2 infection, um, of dementia or Parkinson, but also other kind of pathologies like cancers. Um, we know that the spike protein, through mTOR activation, can reactivate latent viruses. And sometimes these latent viruses are carcinogenic, like EBV for Epstein-Barr virus. Um, so some of, I personally know one case of a lymphoma development in a 17 years old child. child. Um, I know that the amyloid properties of the spike protein are directly linked to its carcinogenic properties. What I explained about RAGE HMGB1 earlier is also implicated in the cancer development. Okay, we have um, a parallel between the amyloid properties of the spike protein and its carcinogenic property. So it's not only degenerative disease we will face, but also cancer. That's quite um,
It's quite that's quite a portrait. Um, so I would love for you to expand on the avenues that you see are the most hopeful to either research further,
Mhm.
um, investigate, or for people who are listening to this and thinking, well, that's beyond the size of a cliff. So, is there anything at all that can be neuroprotective in what you've seen, or has as hopeful, is hopefully if investigated further, if tested, if put in place, could be actually hopeful for people, and it's something that they could easily reach as well, not some molecular thing that's only available in very specialized clinics.
Uh, I think in this context of RAGE HNGB1 system, the better way to counteract this system can be by stimulating SIRT1 in mitochondria and importing niacin. Niacin is also called as vitamin B3 to strengthen the system. Um, if RAGE and HMGB1 team up to promote inflammation and tumor growth, okay, or even trigger degenerative disease, as we discussed previously, SIRT1 is like a maintenance worker in our cells. Maintenance worker. I really apologize. Maintenance worker in our cell. It helps keep things running smoothly by reducing inflammation, for example. Think of it as a cleaner that tidies up messes caused by stress and damage. And SIRT1 can, in fact, dampen the inflammatory signals that RAGE and HMGB1 turn on. And since mitochondria are often damaged in cancer and inflammation, SIRT1 helps protect them. And health mitochondria means less stress and damage, which reduces the release of HMGB1 in the first place. SIRT1 can also help maintain p53 activity. This counters the negative effect of RAGE and HMGB1 on cell growth. So niacin is like fuel for SIRT1. SIRT1 needs an AD plus, a molecule made from niacin, to work effectively. So by importing more niacin, we are essentially giving SIRT1 more energy to do its job correctly. Um, it's more holistic approach than focused therapy. Okay. I think also that some scientists works on icarin. Icarin, it's made, it's take from plants. And also it can counteract the spikes effect. Some scientists, well, are exploring. Some scientists are exploring natural compounds like icarin from plants. Okay, that might protect mitochondria also, and thus counteract the spike protein's effect on the cancer as well, but also on the neurodegenerative disorders. We have also aspirin for the coagulation system that will reduce the platelet activation during the infection, but not for everybody. And we have also, if I remember, I took some notes to forget nothing. Well, I think also I think we have something to investigate about some chaperone protein like HSP70. I think that's something as HSP70 provides protection against misfolded protein, even in Creutzfeldt-Jakob disease, uh, and it either refolds the misfolded protein into the correct shape or tags them for destruction in the cell's cleanup system called proteasome. I think that there is probably something to investigate to heal people in this direction. That's all for today. I don't have much. Eventually, curcumin that could cut the inflammation processes eventually.
Are you aware of any large scale either research hub or hospital or clinic around the world that are working actively on creating actual treatment that um, can essentially reverse these effects?
No,
Are you aware of any? I'm also not. I mean, there's sporadic people, but I'm not aware of a global push at a national level or international level, even less. Like WHO has not mentioned this at all. Like, there's no institution talking about this at
no.
all.
We need today international collaboration between scientists first, then between scientists and physicians. To firstly, we had to understand all the molecular processes to not make mistakes in the treatment. Okay. We have also to develop some diagnostic tools. We had to understand what is happening in the patients. Some patients will present hypersensitivity reactions to the spike protein, that like allergies, okay, with mast cells activation,
Yeah.
something like that. Other
Yeah.
will show uh, more degenerative disorders with the formation of fibrinoids, for example. Other will have brain injuries, etc. So we have to understand what is happening in the patient. We have first to develop diagnostic tools like anti-spike immunoglobulin E the antibodies, okay, or um, to detect regulatory T cells expansion in if the SARS-CoV-2 behaves like HIV, we can face regulatory T cells expansions like in HIV. We have to track the spike protein in tissues. We have to track spike in the blood, where it is. We have to track the viral reservoirs. Okay. But that needs a really huge and international scientific collaboration with many different scientists, um, just immunologists, physiopathologists, etc. Okay, or also probably biophysics to understand where the nanoparticles um, are going in the body.
Distribute.
Okay,
Yeah.
so it's really important for us to understand that we have to connect each other firstly and
Hmm.
exchange in a simple ways with respect to um, to explain our different point of views. Sometimes we can disagree with some theory, but we have to push our arguments in front of the debate. So there is a lot of work.
And at the scale of Europe, so I am depending on where the audience is based. It's going to be people based in North America, but there's also some people based in Europe. What is your sensation about the European medical body, I suppose, and the scientific body and the public health bodies in their actions and role right now?
In Europe, in France, because I'm in France, they are completely disconnected to the reality. Physicians don't know how, what, and where to search the spike protein. I don't even know if they are conscious about the pathological mechanism induced by the spike protein. Don't know. Um, there is also a lot of censorship between all the contributors of the health system. There is a huge censorship. Um, I don't know. I think we have to face two two parts of this of the systemic challenge. Firstly, to connect each other in an international scale, but also in a national scale, and it's quite complicated to perform today. Everyone, everybody wants to be right. Okay, that's not a good way to exchange and discuss the different scientific theory. We have to be firstly humble. I think that scientists or some people, I don't know who, but some people want to be God. We are not God. Firstly, be humble, just to open the discussions, open the connections in order to work together. Firstly.
But in Europe, I'm questioning this because I'm seeing the scene in the US, and it's very disheartening. So I'm wondering whether what is going on in Europe that you think is creating this consensus of silence, which is either we pretend we don't understand, we don't understand,
We don't want
we
to understand.
don't want to understand, we don't want to do anything about it if we do understand. So is it because we are captured in terms of like the reach of pharmaceutical inside the very big bodies of public health
Mm.
and research in France are actually just as eaten as they are in the United States? Or do you think this is a very European specific problem that's making this
Mm.
willful blindness at scale?
I think we are all cloisoned in some boxes, and each actor in this crisis is working into its own box. Um, that's why I told you, I told you, we have to connect each other firstly. Um, we have to be more open-minded. I think. I think it's not specific to Europe. It's not specific to America. It's not specific um, in many different countries. What can I say is that everyone wants to be it own master in its field.
But isn't that playing into the hands of those who pay them? So, in other words, um, given what we're seeing in the United States, it seems almost impossible to change something at the depth of the system because the system is funded, pushed, architected by such enormous powers in terms of financial power, pharmaceutical power, etc. So, what about Europe? Is it just the same? Is it the case that Europe is also extremely eaten by pharmaceutical company
Yeah,
in science, in publication, in training of doctors, in training of scientists, and therefore that these boxes that we all end up in are actually very useful boxes for those people because
they can control
at this,
everything.
they can. But I'm wondering, same as I did during COVID. I mean, it's a real, it's an honest question. I'm just not asking this for rhetorical reason. I was stunned by the fact that as physician got given those injections, they didn't understand them because nobody did, apart from if you work for BARDA or Gain of Function or Eco Health Alliance or whatever. But I think at the scale of every physician, let's say in the UK, I saw it. You saw it in France. I saw it in various countries in Europe applying that. You get this thing. You have most of these insert were empty in this pockets of the vials themselves. You don't understand any of this, but you're going to go behind this message, which is I've been told this is safe and effective. I will carry that message safe and effective, and I will then advise you to get it. And then there seem to have been a plethora of people who went along that road, a few that didn't, and they lost their license, or they got sued or whatever it is, or they disappeared or whatever. But the vast majority went along and went behind it, and now continue to be silent about that. And in the so that's the clinical part. In the science part, there's equally amount of people that just went silent. They made no comment about the mRNA. They made no comment about gain of function. They make no comment about the virus itself as a potential bioweapon. So, what is that in your mind? What is that? The total silence of something that you are equipped to understand that you don't understand. Therefore, you should say, I don't understand it. I can't get behind it. All I can do is, I've been told to give you this. I don't get it. I don't want to give you this, but I have to give you this. If more people had just even said that, don't you think that stuff would have stopped? Like, what is that? What is the silence thing?
Oh, that's quite complicated to explain in English. I think that some people were afraid by the virus. I think. I think. Firstly, we didn't understand anything at the beginning. Okay, uh, we didn't even know where the virus came from. Okay, um, so the physicians, I think, made their best to treat their patients. Firstly, but that was quite complicated for them to perform their own job. Okay. And then after that, they pushed the anti COVID-19 injections. Okay. But now, how can you admit that you were wrong when you know that the SARS-CoV-2 can behave like HIV? Okay. And the anti COVID-19 injection can reinforce the virus in the body. It's quite complicated to admit that you were wrong. We have first to change our mentality. Everything that was really complicated during all this crisis. Everything was really complicated. Treatments were complicated. The I don't know how to say that in English. Les traitements étaient compliqués.
Treatment were complicated.
Les mesures gouvernementales étaient assez sévères.
The government measures were very serious and very harsh.
Yes. Et aujourd'hui, comment, avec tout ce qui a été fait, peut-on dire que nous avions eu tort?
And how can we say today, with all the push that was done, that we were wrong?
Nous devons d'abord commencer par changer les mentalités. Se dire être humble. Dire, OK, on s'est planté.
We must first change our mindset and accept that we got it wrong. But I think that's exactly what I'm getting to. Is I think there is this revealed an enormous problem in the medical profession and in the scientific domain profession, that your critical thinking and your personal ethics and your personal approach to this problem, it was never going to be about you. It was never going to be about clinicians. It was never going to be about scientists. What was needed at that point is for them to be human first, critical thinker first, humble first, and then to apply their skill set to help us get through this. And in fact, what they did is they put this huge theme as a cult, like, I'm a scientist, I'm an expert, I'm a medical doctor.
I have all the badges.
I have all the ticks, the badges, I got my CVs ready. You don't know anything, shut your mouth. Get it done. And to me, this we have in front of us a enormous job to rebuild the scientific mindset, education, whatever it is, and the clinical relationship. And I just wonder whether that's even in the plans, because right now they're not even recognizing at scale that they did everything wrong.
Yeah.
They managed this in a most hideous way, that people failed. They failed other people. You can say, you said the word humble. Where do we hear every day in Europe people coming through and saying, got this wrong, didn't understand that, was too late to that, misunderstood this. I'm so sorry I did that to you. Nowhere. It doesn't. It's not happening.
And it will not happen. It's really, it's là, on est plus dans l'ordre carrément, en fait, de la conscience. Et
Yes. Yeah, we are in the realm of consciousness and ethics. But you when you join the medical profession and when you join the scientific community, you have an oath. In medicine, you have an oath.
not in science.
Not in science. That's a very good point.
Not in
And I
science.
think you should. And I think you should.
Normally, yes, but no.
I know.
We,
But even when they had the oath, they threw it out the window.
yeah, complicated.
So that in and of itself, this is a super scary realization. I know you are a professor. You teach. You train young minds. I think that should be the forefront of the change. That the the mind makers should not just be brutally throwing down the right type of science, the right type of thinking, but rather, do you have the right mindset?
I sometimes, I'm telling to my students that we always need a smaller person than you. That's not because you made a lot of students that you know everything. That's all. And it's not because the person in front of you has not your diploma, your diploma, uh, that he is wrong in his words. You have firstly to listen what what the people, the smallest people is telling firstly, understand his words and think about these words and consider it not as an enemy, but as a partner of your thinking. And I love my students because sometimes they want to challenge me um, on my knowledge. And it's quite funny because that's really stimulating. That's really stimulating, really.
Mm,
And it's a big exchange. That can be a big exchange if I put myself on their level.
mhm,
But only that. And after that, you know, I don't think we can fight the system. We have first to change ourselves. We cannot fight the system
mm,
personally. What can you do? You're we are isolated, alone. We are working on our side. So you cannot change the system by your own. Impossible. But
mm,
if we all together change our mind and try to work together, not taking words um, in terms of um, harmful words, but taking the words as partners of your thinking. I don't know if I express myself correctly, but
mhm.
it's quite complicated to formulate.
It's
Mm,
it's like the Sisyphean task. You push that boulder up the hill, and you hope that no one else pushes it forward, back down, because we're all carrying that boulder up. And I think it bears repeat that nothing is achieved alone, but by the same token, every one of us has a responsibility to be the best person they can be with the skill set they've got. And the more skill you have, the more responsibility you have. That's my view. So, with that, I would like you to tell the audience where they can find your work. We will put a lot of the links in notes of the this particular episode. But please let us know where they can find your work.
on X, essentially on X. Um, well, I work a lot on immunology.
Hmm.
Okay, I'm I'm writing some thread that I post on X, but also on Curious with my article. And now I'm really I have to write the second article I want to publish on the immune tolerance mechanism and my theory that SARS-CoV-2 behaves like an HIV and uh, a prionogenic HIV.
That would be probably another episode. So, Annelise, to conclude, I will ask you the question I ask all my guests, which is, what is your weird sauce that powers your life, your hopes, and your work? What is the special sauce that gets you going?
Kindness, I don't know.
That's a great one.
That's mine. More kindness
Excellent.
in the world. Thank you.
And Bouquet, thank you very much for your time. And we will catch up with you and your work in the future. In the meantime, people can check all the links in the show notes and follow all your work on X, as well as follow all your work in the Francophone areas, which is where you mostly operate. But thank you for making the effort of delivering this podcast in English.
Thank you very much to you.
If this conversation stopped you in your track, share it with your network. You never know whose life you might change for the better. Thank you for listening. Stay curious about our next guest and stay curious about life.